If you've ever worn a cheap piece of nickel jewelry or maybe accidentally brushed your ankle against poison ivy while hiking, you know exactly what happens next.
Oh, absolutely. It's unforgettable.
Right. First comes the redness, then the swelling, and finally, that unbearable, just all-consuming urge to scratch.
Yeah, an urge so strong that you will literally tear your own skin apart just to get like a single moment of relief.
Exactly. And historically, if you went to a doctor for that, the explanation was pretty straightforward, right?
Yeah, the classic textbook answer is that your immune system is overreacting to a harmless chemical, and your nerve endings are simply feeling the damage and sending an ouch or itch signal up to your brain.
Right. The nerve is just a dumb wire. It's a sensor acting like a smoke alarm reporting a fire.
Precisely, just a passive reporter.
But what if that wire isn't just reporting the fire? What if the wire is actually controlling the fireman? Okay, let's unpack this. Today, we're doing a deep dive into the hidden architecture of the nervous system.
And it is a fascinating architecture.
It really is. Specifically, we're unpacking a groundbreaking 2026 paper from the journal Immunity by Voisin and colleagues. Our mission today is to explore how our nerves independently control the two most miserable symptoms of skin allergies, the itch and the swelling.
To really understand how revolutionary this paper is, we have to look at a condition called allergic contact dermatitis, or ACD.
Which is super common. All right.
Oh, incredibly common. It affects up to 20% of people. And to study exactly how this allergy develops in the tissue, researchers rely on a highly specific biological model in mice called contact hypersensitivity, or CHS for short.
Okay, CHS. But how do you guarantee a mouse gets an allergic reaction on command? Like, you can't just rub poison ivy on them and hope for the best.
No, you can't. You use a synthetic chemical allergen called DNFB. The full name is 1,0,4-dinitrofluorobenzene, but DNFB is the standard abbreviation.
DNFB, got it.
And the timeline here is critical because an allergy isn't an instant reaction, you know. It's a learned immune response.
Right, your body has to recognize it first.
Exactly. So at day -5, researchers apply the chemical to the mouse's abdomen. This doesn't cause a rash. It simply introduces the chemical to the immune system. Specifically, local immune cells pick up the chemical, travel to the lymph nodes, and show it to the T cells, priming the body's defenses.
It's like putting a suspect's photo on the immunological most wanted list.
That is a perfect way to describe it. Then they wait 5 days. That's the incubation period. At day 0, they challenge the mouse.
By putting the chemical on them again.
Yes, applying it again, but this time directly to the skin on the ear or the back. Because the immune system is primed and ready, the reaction is fierce. Over the next 4 days, day 1 through day 4, the researchers observed severe tissue inflammation and intense scratching.
Okay, so we've got this chemical sitting on the skin and an immune system going haywire. But what is physically sensing that chemical? I mean, my skin itself doesn't have a brain.
It comes down to a specialized class of neurons called nociceptors. These are your pain and itch-sensing nerves.
Nociceptors.
Yeah. I want you to visualize your spinal cord as this massive, high-speed fiber optic trunk line running down your back. Just outside of that trunk line, at every single vertebra, you have these little nodules called dorsal root ganglia, or DRGs.
I always picture the DRG as a secure off-site server room.
That's a great analogy.
Yeah, like the main the cell body of the nerve sits safely in that room, and it runs a single, incredibly long microscopic wire all the way down your arm or your leg, right into the top layers of your skin.
Exactly right. That's a pseudononopolar neuron. And for decades, if you looked at those wires under a standard microscope, they all looked pretty much identical.
Just a bunch of identical cables.
Right. But they aren't. When we look at their active genetics, their transcriptomic profiles, we find that nociceptors actually come in 6 distinct subtypes.
6 of them, wow.
Yeah, they're categorized broadly into 2 families. First is the peptidergic family, which we'll call PEP nerves, and then the non-peptidergic family, which we'll call NP nerves.
Okay, so PEP and NP, what are the actual 6 types?
So in the PEP family, you have PEP1, which expresses the genes TRPV1 and TAC1. Then there's PEP2, which expresses TRPV1 and NEF.
Okay, so that's 2.
Then in the NP family, you have NP1, defined by NBPRD. You have NP2, which expresses MERGPR3, and NP3, which expresses SINCT.
That's 5. What's the last one?
The last major one is the TRPM8 subtype, which is famous for sensing cold.
Okay, so we have these 6 different flavors of pain sensors parked in the skin. If I'm thinking about this logically, if I'm having a severe allergic reaction and my skin is red and screaming with an itch,
Right, the typical response.
if I just cut the power to those sensory nerves, the whole thing should calm down,
Yeah.
right? Like I wouldn't feel the itch and maybe the stress on the tissue drops.
You would absolutely think so. It makes total sense. And that is exactly what the researchers tested first in this paper.
Oh, I did.
Yeah, they used a genetic model called NaV1.8 IDTA.
Okay, that sounds like a password. What does that mean?
It's a mouse engineered to be born entirely without these specific pain-sensing nociceptors. Basically, the NaV1.8 gene is unique to these pain nerves. So they rigged it so that any cell expressing NaV1.8 also produces a toxin, the IDTA, that kills the cell during development.
Oh, wow. So the nerves for light touch are totally fine, but the pain and itch sensors are simply gone from birth.
Exactly.
It's literally like going to the breaker box and cutting the main line to the alarm system.
Yeah.
So what happened when they exposed these mice to the allergen? Did the reaction stop?
Well, they discovered a massive biological paradox. As expected, the mice completely stopped itching, zero scratching.
Okay, that makes sense. The itch wire is cut.
Right. But their skin inflammation didn't calm down. In fact, it exploded.
Wait, exploded?
Yes. The swelling was vastly worse than in normal mice. The skin thickened drastically, and the tissue became completely overrun with a type of immune cell called a neutrophil.
Wait. So turning off the pain sensors made the swelling worse?
Significantly worse. What's fascinating here is that this proves nociceptors are not just one-way streets. They aren't just sending an alarm signal up to the brain.
They're doing something else.
They are actively broadcasting signals back down into the tissue to control the local immune response. The nerves are acting as the brakes on your immune system.
Okay, let's do a quick recap here to make sure I'm wrapping my head around this.
Go for it.
So nociceptors live in that DRG server room. They come in 6 distinct flavors. And turning them all off stops the itch completely, but causes severe runaway inflammation.
That is the perfect summary.
So for you listening, let me ask you this. Have you ever considered that your nerve endings might be the only things preventing an allergic reaction from spiraling completely out of control?
It really flips the script, doesn't it?
It changes everything about how I view a rash. But it also creates a massive mystery. If turning off all the nerves stops the itch but makes the swelling worse.
How do we know which nerve does what?
Exactly. How do we know which of those 6 flavors is driving the itch and which one is hitting the brakes?
That was the next major hurdle. The researchers had to figure out a way to map the plumbing, so to speak. They needed to isolate only the nociceptors that physically ended in the skin, completely ignoring the ones that go to your stomach or your muscles.
Right, because the DRG server room sends wires everywhere, not just the skin.
Exactly. To do this, they used a technique called retrograde tracing.
Retrograde tracing, how does that actually work?
They inject a specialized fluorescent dye, specifically one called DI, right into the skin of the mouse.
Okay.
That dye gets picked up by the microscopic nerve endings and physically travels backward, up the long axon, all the way back to the DRG server room next to the spinal cord.
It's like pouring glowing neon paint down a specific drain in your kitchen, walking down to the basement and seeing exactly which pipe lights up.
Yes. And once the dye arrives, they extract the DRG and use a laser sorting machine to pull out only the individual nerve cells that are glowing neon.
That is incredible. Okay, so once they have those specific glowing skin pipe cells, what are they looking for?
They look at the active genetics of those individual cells using a technique called single-cell RNA sequencing, or cRNA-seq.
Okay, so breaking that down, RNA is essentially the active blueprint a cell is reading at any given moment, right?
Right. If DNA is the whole library, RNA is the specific book the cell has open on the desk.
Got it. So what did the blueprint say?
They found that the deep tissue nerves were mostly from the PEP family, but the nerves terminating right at the surface of the skin, those were overwhelmingly from the NP family, specifically NP1 and NP2.
Interesting. So the skin is mostly covered by these NP wires.
Yes. And even more surprising, these skin nerves had specific receptors like TRPA1 that allowed them to directly taste the DNFB chemical allergen themselves.
Wait, really? So they didn't need the immune system to tell them there was a problem?
Nope. The nerve felt the chemical directly.
But the allergy isn't just a static event. Like we said earlier, the reaction builds from day 1 to day 4. Did they look at what those NP nerves were doing as the rash got progressively worse?
They did. And this is where the data gets incredibly strange. When they looked at the active genetics of the skin nerves at the peak of the reaction at day 4, They found a mysterious new group of neurons that didn't fit into the normal 6 categories at all.
A new group.
Yeah. They called it the ND cluster for not determined. At day 1 of the allergy, it barely existed. But by day 4, it had massively expanded to make up nearly 22.5% of all the pain nerves in the skin.
Wow, almost a quarter of them. And here's where it gets really interesting. When they looked at the active blueprints of this mystery ND cluster, they saw a specific gene lighting up called ATF3.
Yes, ATF3, along with a few others like GAL and SPAR1A. These are what biologists call regeneration-associated genes.
Regeneration-associated genes, so like emergency response genes.
Exactly. We normally only ever see AT3 turn on when a nerve has been physically crushed or severed with a literal scalpel. It's the nerve's internal toolkit for rebuilding a severed axon.
But nobody took a scalpel to this mouse. It's just a chemical rash on the surface of the skin. Why would a nerve firmly believe it's just been amputated?
Because of the collateral damage. An intense allergic reaction isn't just red skin, it's chemical warfare. The immune system is flooding the area with toxins to destroy what it thinks is a threat, and the surrounding tissue is getting destroyed in the crossfire.
So the environment is just highly toxic.
Highly toxic. That harsh environment physically degrades and damages the microscopic nerve endings. It's effectively a severe chemical burn.
Okay, so the nerve is getting chemically burned. But nerves in adults don't typically just multiply, right? You don't just grow 25% more nerves in 4 days. Where did this mysterious ND cluster actually come from?
That was the big question. To answer that, the researchers used a complex computational algorithm called Monocle 3. It performs something called pseudotime analysis.
I've heard of this. You take all the single-cell RNA barcodes from thousands of cells, and the algorithm plots them on a massive graph to find the relationships.
Exactly.
It's like finding a million individual scrambled frames of a movie scattered on a cutting room floor and having an AI arrange them into a perfect flipbook so you can watch a cell physically change its identity over time.
That flipbook provided a definitive answer. The mystery cells didn't come from nowhere. They were the NP1 nerves.
The normal skin nerves.
Right. The NP1 nerves in the skin had been so damaged by the allergic inflammation that they transcriptomically reprogrammed themselves. They rewrote their own internal operating system, shifting from a normal resting state into a frantic wound healing state characterized by that ATF3 gene.
Okay, let's do another quick recap. Skin nerves are mostly NP1 and NP2 types. During a severe allergy attack, those NP1 nerves rewrite their own code to look like they've been physically injured, expressing the ATF3 gene.
Spot on.
So for you listening, think about this. If a nerve thinks it's been cut just because of an allergy, what kind of signals is it sending to your brain?
It's a huge question. Does it feel like a burn, or is that the itch?
That's exactly what I want to know. We have this shape-shifting NP1 nerve that is turned into an ATF3 nerve. Is it the thing driving the urge to scratch, or is it the brake pedal holding back the swelling?
To figure this out, researchers had to get incredibly precise. They used 2 highly targeted genetic models to selectively delete only the nerves that expressed ATF3.
But how do you delete a specific nerve halfway through an experiment without messing up the rest of the body?
You use a chemical lock and key. One of the models they used was called ATF3 T2 IDTA.
Okay, another password. Break that down for us.
So the mice were engineered so that any cell expressing ATF3 also contained the instructions for a lethal diphtheria toxin, the IDTA.
Okay, a suicide switch.
Right. But those instructions were locked safely outside the cell's nucleus by a protein called ERN2. The lethal instructions could only enter the nucleus and trigger the toxin if the researchers gave the mouse a specific drug called tamoxifen.
So it's like a Trojan horse that only opens when you send a very specific chemical signal.
Exactly. So they let the severe allergy develop for 4 days. They let the NP1 nerves reprogram into AT3 nerves. And then right at the peak of the allergy, they gave the tamoxifen.
What did that do?
It selectively assassinated only the nerves that had reprogrammed, leaving all the healthy, normal nerves completely untouched. They also confirmed this with another depletion model called GNP-HETR, which targets the same lineage, and the results were identical.
And what were the results? What happened when the reprogrammed nerves died?
The itch vanished completely.
So gone.
Gone. But the inflammation, the red, swollen skin full of immune cells, stayed exactly the same as a normal allergic mouse.
Wow. So those shape-shifting NP1 nerves are the dedicated itch switch. They drive the urge to scratch, but they have absolutely zero control over the actual swelling in the tissue.
That's the conclusion. And it provides an incredibly compelling new mechanism for humans, too. Think about chronic eczema or unexplained chronic itch. We often treat it as a surface-level skin issue.
Right, with topical steroids or moisturizers.
But this research suggests that chronic itch might actually be a misguided wound healing response deep within the nervous system. The nerve gets damaged by chronic inflammation, shifts into this regenerative ATF3 state, and your brain interprets that specific distress signal not as pain, but as an overwhelming urge to scratch.
That is wild. But hold on, I need to push back here for a second.
Sure.
If we definitively prove that the NP1 nerve drives the itch, but does absolutely nothing to the swelling, what was acting as the brakes in that very first experiment? Because remember, when we cut the main power line to all the pain sensors, the swelling exploded. If NP1 isn't the brakes, what is the fire extinguisher?
To find the fire extinguisher, we have to look back at the PEP family of nerves, specifically the PEP1 subtype. These neurons are famous for expressing a receptor called TRPV1.
TRPV1, I know that one. That's the receptor that senses heat, right? It's the one that makes hot peppers taste spicy.
Yes, exactly. And because they knew that, researchers didn't use a genetic trick this time. They used a chemical ablation. They injected the mice with a biological compound called resiniferatoxin, or RTX.
RTX, that's essentially ultracapsaicin, right? Like the active ingredient in pepper spray,
Yep.
but millions of times more potent.
It is unbelievably potent. It binds so aggressively to that heat-sensing TRPV1 receptor that it completely overloads and burns out the specific PEP1 nerves. It physically destroys them.
Oh, wow.
But here's the key. It leaves the itch-causing NP1 nerves perfectly intact because NP1 nerves don't have that heat receptor.
Okay, so now we have a mouse with a fully functioning NP1 itch circuit, but its TRPV1 heat circuit has been burned away. What happens when you expose this specific mouse to the DNFB allergen?
The mice scratch normally. Their itch circuit worked perfectly.
Because NP1 was still there.
Right. But their skin became catastrophically inflamed. The tissue was completely overrun with neutrophils, far, far worse than a normal reaction.
So the TRPV1 nerves are the brakes.
Yeah.
They're actively holding back the swelling. But you keep mentioning neutrophils. Why are neutrophils specifically the problem here?
Well, neutrophils are essentially the immune system's SWAT team. They are first responders, and their job is to destroy foreign invaders, but they are messy.
Messy how?
They release enzymes and reactive oxygen species that cause massive collateral damage to your own healthy tissue. To prove that neutrophils were the actual culprits causing the severe damage when the nerve brakes were removed, the researchers did one final rescue experiment.
How do you rescue a tissue from a microscopic SWAT team?
You take away their transport. The researchers took the mice missing their TRPV1 nerves, the ones with runaway inflammation, and injected them with a specific antibody called anti-Ly6G.
Anti-Ly6G, what does that do?
This antibody actively hunts down and depletes neutrophils in the bloodstream before they can ever reach the skin.
And without the neutrophils arriving at the scene?
The severe runaway tissue damage completely reversed.
That's unbelievable.
Yeah, it proved definitively that in a normal, healthy state, your TRPV1 nerve endings are constantly monitoring your skin, communicating with your immune system, and actively suppressing those neutrophils, keeping them from flooding the area and destroying your own tissue.
Okay, let's lock this in for the listener. I've got a quick memory trick for this. Just remember N and TI.
N and TI. I like it.
Yeah, so itch is driven by NP1, that's IN, and TRPV1 stops inflammation, that's TI. So NP1 nerves shift into an ATF3 state and cause itch. TRPV1 nerves act as the brakes on neutrophils, preventing runaway inflammation. And they operate totally independently.
Completely independently. It's a stunning separation of duties.
So if you're listening, think about the practical application of this. If we can target these circuits independently, could we create a topical cream that completely turns off the urge to scratch without interfering with the immune system's actual work?
That's the dream. We wouldn't have to nuke the whole immune system with heavy steroids just to stop the itch.
It's just wild. We have two totally distinct systems operating side by side in the exact same millimeter of skin. On one side, NP1 nerves sensing damage, shape-shifting, and making a scratch. On the other side, TRPV1 nerves acting as local peacekeepers, telling the immune system's SWAT team to stand down.
If we connect this to the bigger picture, this research really shatters the old textbook model of neurobiology.
It really does.
The old model was highly centralized, right? The immune system does all the complex cellular work locally, and the peripheral nerves are just passive, dumb wires sending a status update to the brain. But this paper proves that our peripheral nervous system is a highly sophisticated, localized control board. It manages host defense, the immune system, and behavioral responses like scratching as two completely distinct programmable circuits.
It makes you realize how little we actually knew about the wiring of our own skin. It's not just a wrapper keeping our insides in, it's the dynamic computational organ.
Well, thank you all for taking this incredibly dense, really detailed journey through the transcriptomics and neurobiology of allergic contact dermatitis with us.
It's complex stuff, but so important.
Absolutely. And to wrap up our deep dive today, I want to leave you with one final thought to mull over. Yeah, and it's a profound one. For decades, modern medicine has treated complex inflammatory conditions, things like severe eczema, psoriasis, or even internal issues like inflammatory bowel disease, almost exclusively by targeting immune cells. We prescribe heavy biologics to artificially suppress the white blood cells.
But if our nerves are actually the puppet masters?
Exactly. If our nerves are dictating exactly where the immune cells are allowed to go, acting as the local brakes and independently rewiring their own genetics to alter our behavior, what other devastating autoimmune diseases are actually just nervous system glitches? What if the immune system is perfectly healthy, but the nerve holding the leash has simply let go?
Think about that the next time you feel a random itch. The whole ecosystem in your skin is communicating right now. Keep diving deep.